snakes are found in every type of habitat. They are capable of bringing down prey animals which are almost 100 times larger than themselves. Venom is probably one of the key factors that makes them such successful predators and conquer almost every patch of land on Earth. So if you look at snake venoms, they are very complex. They contain hundreds or thousands of proteins. But what is most fascinating about this is that many of these proteins are also found in you and me. So the first thing that came into my head was, how do these completely non-toxic proteins that are so important for our survival have become some of the most potent toxins that we have known. That's when I realized that this is what I want to study for the rest of my life. I'm Kartik Sunagar, an evolutionary biologist working as an associate professor at the Indian Institute of Science, Bengaluru. My lab mostly works on venoms, snake venoms, and designing better antidotes for treating snake bites. Snakebite is a poor man's problem, and India is the snakebite capital of the world. In India alone, snakebite kills over 58,000 people. 3 times that number suffer permanent disabilities, which for most is worse than death. So snakebites are treated using antivenoms, which are manufactured by collecting venoms from snakes. So there are lots of issues with current antivenoms. One is that they are being manufactured using a technology that is over 100 years old. So the result is that you have very inefficient antibodies that neutralize snake bites. This antivenom is also produced in horses by immunizing horses and other animals, which causes them a lot of pain. And this antivenom doesn't work across geographies. So the conventional antivenoms are produced to treat bites from 4 species called as India's big 4 snakes. These are spectacle cobras, common krait, saw-scaled viper, and Russell's viper. But there are many other species of snakes that are found in pockets of India. For example, if you go to eastern part of India, you will encounter monocellate cobras. And if you're bitten by those snakes, you don't have a specific drug. You are treated with the conventional antivenom. We have also discovered one of the most toxic snake in the country. It's probably the 3rd most toxic snake in the world. Originally, it was found only in the Sindh region of India and Pakistan. It is called the Sindh krait, but now recently we have discovered even in the northern part of Karnataka. So if you are bitten by this snake, again there is no specific treatment. If you are walking in the Western Ghat, you could encounter king cobras, you could get bitten by one of the pit vipers. Same story with snakes in Northeast India. So no matter which other species of snake bites you, the clinicians have no other option but to treat those bites using the big 4 antivenom. A single antivenom cannot neutralize all of this variation in venoms. So this is where our research comes in. So when I started this lab, we started going out to the field and collecting venoms of snakes from the wild. Cobras, for example, when they bite, your body is unable to breathe because neuromuscular receptors get blocked. So you are unable to breathe, and that's why people die. But in the neighboring states of the very same populations, we saw that the cobras, they were causing a lot of necrosis, meaning that if you were to bitten by that snake, then you would lose your arm or limb, depending on where it has bitten. So that was very surprising that even populations that are next to each other can produce very different venoms. tested our Indian antivenoms, whether they are capable of neutralizing these venoms, we realized that they poorly neutralize the venoms of most individuals. So the outcome of snakebite treatment can also depend on which individual snake bit you. So this, perhaps I would say, was the most surprising of all of our findings. We are actually trying to address this problem by various means. One is that we are trying to produce antibodies in the lab that target specific toxins in snake venoms, and these antibodies are capable of neutralizing venoms across geographies. They are also produced in cell lines, so you don't have to cause animal harm to produce them. The other way is by introducing these orally consumable pills, which again target some of these key toxins in snake venoms. Imagine that you're bitten by a snake while walking in a forest. You can just reach out to your pocket and take one of these pills. It either circumvents the need for taking antivenoms altogether or gives you a lot of time to reach the closest hospital that is trained to treat snake bites. So another approach we are taking is by discovering antibodies from synthetic libraries. So imagine that I take your blood, I isolate all the cells that are producing antibodies in your body, I mutate them further to generate a huge combination of antibodies that don't even exist in nature. Now what I can do is I can select key toxins from snakes and use them as baits to fish out antibodies that are very specific to those toxins. So when I do, we can sequence those cells and we can then start producing your antibodies in the lab itself. And these antibodies are called recombinant antibodies, which can then be used for treating snakebite because they neutralize some of the most important components in snake venoms. One of the antibodies that we discovered using this approach was capable of neutralizing monocellate cobras in eastern India, black mambas in sub-Saharan Africa, and one of the most toxic kraits in Southeast Asia. So we demonstrated that you can produce these synthetic antibodies in the lab that are capable of completely neutralizing venoms of some of the most deadliest snakes that are ever known to us. So probably the most happiest moment for our research was discovering this antibody against Russell's vipers. Russell's viper is probably responsible for over 50% of all snake bites in India, and it is considered the most medically relevant snake in the world because no other snake species kills and maims as many people as this species does. And our understanding changed quite a bit when we realized that while they have hundreds of different toxins, they all work in synergy, they all work in a concert. So if you break that circuit, if you break that partnership, you will be able to neutralize the venom completely. So we found that a single antibody made in the lab was capable of neutralizing venoms of Russell's vipers in every corner of India. So venom research in India has started almost 50 years ago. I started this lab about 8 years ago, and since then we have been striving hard to introduce these next generation of snakebite therapies for India and elsewhere. But unfortunately, it will take a lot more time to see this translation from bench side to bedside. Right? And that's mostly because of the bureaucratic hurdles that we often face, be it with the time that it requires to obtain permissions to collect snake venoms or to test our medications in human clinical trials. For example, when the world was hit with this pandemic called COVID-19, right? So the vaccines were basically made available to people in less than a year. And all of this happened because it was a global emergency. So there are real world examples that things like this can be done, and snakebite is a major health crisis that is probably one of the most neglected. So if the government supports our research and helps us to test these drugs in human clinical trials, then I think we will be able to see these drugs a lot more sooner in the market.